So in this time, I'd like to talk about the acupuncture modulates brain circuits to alleviate emotional and cognitive dysfunctions in drug addiction and withdrawal model. Today's presentation will follow this content. The first time the introduction. In the introduction session, I will talk about the background of the emotional and cognitive dysfunctions in the drug addiction. And second session, I'd like to talk about the present my experiment result published in the 2019, the effect of acupuncture on alcohol withdrawal and dependence. And last thing is my published in 2021-23 experimental result, the medial prefrontal cortex and lateral habenula and rostral medial tegmental area pathway in the acupuncture effect in cocaine psychomotor response. Everyone well known that the addiction, substance use disorder, commonly known the drug addiction, is medical illness associated with the compulsive and repetitive use of the addictive substance. It's defined especially as compulsive patterns of the drug seeking and drug taking compared to take place at the expense of the most other activities. The key question is the addiction, or why do some susceptible individuals undergo to transition from the casual drug use to compulsive patterns of drug use, and why do addicts find it so difficult to stop using that? In the drug addiction, when the drug addiction will change one part of our brain, in the cell level, they change the neurotransmitter release and change the transcription factor expression and other things in the cell level, it make the molecular network changing. And then it changed the cell and cell communication in our brain nerve system. Finally, it changed the organ system in the animal model, expressed the behavior change in the anxiety and depression and panic disorder and drug addiction. These statistics show that alcohol abuse, drug addiction showed a very high rate of the reinstatement compared in other illness. And in our Korea, show that and now is not clear area of the drug. Show that increased many suggest drug and arrested people. In the arrested people, it showed the office worker and students and doctors were increased steadily. Maybe this data showed then in 2016, so maybe, and now much more increased that the data were maybe increased, I think. So in drug addiction showed some features, the compulsive and repetitive use of the addictive substance, and showed severe withdrawal symptoms and high reinstatement rates. So importance of it showed the importance of the prevention and treatment of drug addiction disorder. In our brain have the four major dopamine pathway. One is the mesolimbic pathway, and neurosteroid pathway, mesocortical pathway, and frontodental ventricular pathway. In among them, the mesolimbic pathway is the major circuit of the drug addiction. So it is mediated motivation and reward or pleasure. So mesolimbic pathway consists with the nucleus accumbens. nucleus accumbens and the ventral tegmental area. It is controlled the dopamine release of in our brain. So when release the dopamine, we feel some motivation and feel some pleasure and reward. So anti-addiction agents target this measuring big pathway, the composite and naltrexone, or control the release of some neurotransmitter, and it is mediated nucleus accumbens and the ventral tegmental area. So it controls the fear of the reward. But these drugs have some common side effects, the anxiety and depression and dizziness and everything. So it is important that is the need to develop the safety and non-invasive treatment method. So we need to in the treatment of the drug addiction. And the research of the drug addiction lead to some animal model, because in the establishment in the animal model need to capture defining the features of the human drug addictive behavior, the escalation and binge use, binge use and enhance the motivation and preference of the reward options and enhance drug seeking relapse. So if you use in the animal model, need some learning and memory of specific situation, take a drug and other things, and observer change the behavior. So need to some rodents or mammals with a high level of cognitive functions. So in this study, the purpose of this study is to identify the neuronal mechanism contributing to acupuncture stimulation, induced inhibition of addiction behavior. So first session is the suppressive effect of acupuncture stimulation on addictive behavior in the alcohol dependence. And the next is the neuronal pathway that conveys acupuncture signals from the peripheral acupoints to the brain. In this study, I used the acute cocaine addictive animal model. The first result is the effect of acupuncture on alcohol withdrawal and dependence. It is published in the Scientific Science Advanced in 2019. In alcohol addiction, the neurobiological substrate of the alcohol addiction in animal and human is believed to some way involve the dopamine system of the nucleus accumbens, and various brain area was mediated alcohol addiction, the arcuate nucleus and nucleus accumbens, arcuate nucleus and arcuate nucleus and nucleus accumbens and ventral tegment area. The beta endorphin and GABA and dopamine signal are associated also. And I described the alcohol dependence. In the arcuate nucleus is a major part of the beta endorphinergic neuron. When the alcohol dependence decrease the beta endorphinergic signal from the arcuate nucleus to nucleus accumbens. So the result really, the GABAergic signal was increased and the dopamine signal was finally decreased. So it makes the hypodopaminergic states. So it can make the increase of the physical withdrawal signs like a tremor, and increase the psychological withdrawal signs, evoke the psychological withdrawal signs the anxiety and depression. So resultantly, it makes the alcohol-seeking behavior in the alcohol dependence. So this result showed the first in the alcohol withdrawal increased the tremor response. And when we treated the acupuncture stimulation, it was significantly decreased the tremor. So we treated the acupuncture at the HT7 and the Shinmun acupoint. And the control acupoint LI5 was not significantly effect of the acupuncture effect in the tremor response. And we detected which neurotransmitter were involved in the acupuncture effect. So we analyzed the microdialysis. I used the microdialysis and we analyzed the change of the neurotransmitter release. So our results show that when we treated the acupuncture was significantly increased the release of the beta endorphin. In the withdrawal model was basal endorphin level was significantly decreased, and when we treated the acupuncture, it showed the increase of the beta endorphin levels. And then we treated the beta endorphins directly in the brain. It showed the decrease of the tremor. So this result means that the acupuncture effect on the withdrawal-induced tremor is maybe mediated the beta endorphin release. And next, we tested the anxiety-like behavior using the elevated pulse maze. Results show that the acupuncture was significantly increased acupuncture was showed decreased anxiety-like behavior and increased the movement of the open arm time. So it is used also when we treated the beta endorphin, and we can show the removal of the anxiety-like behavior. And next, we detected the neuronal circuit of the acupuncture effect. So this research showed that when we treated the acupuncture was first increase the activity of the arcuate nucleus, the major part of the beta endorphin neuron. And when we give some retrograde tracer into the nucleus accumbens, and this red mark is the show the neuronal tie from the arcuate nucleus to the nucleus accumbens. So when we treated acupuncture, this neuron was showed the C-force expressed. It means that this neuron was activated after by the acupuncture stimulation. And we detected the neuronal activity by the extracellular coding. At first, the basal level of the neuronal firing rate was arcuate nucleus is decreased during the alcohol addiction, but when we treated acupuncture, this neuronal activity was significantly increased compared to control. And next, we detected the change of the dopamine expression in the ventral tegment area. So we used the immunochemistry and detected the TH, tyrosine hydroxylase expression. So when we treated the acupuncture, we treated the beta endorphin, showed that the TH expression was significantly increased. So this result showed that the effect of acupuncture was increased the activity of the arcuate nucleus, and we improved the release of the beta endorphin, and resultantly it changed the dopamine release activity. So finally, we checked the ethanol-seeking behavior when we treated the acupuncture at the HD7 was significantly decreased the ethanol-seeking behavior. And also when we treated the beta-endorphin was significantly decreased the ethanol-seeking behavior. So summarize that when we treated the acupuncture at the HD7, we increased the arcuate nucleus activity and increased the beta-endorphin release. And finally, it improved the dopamine depletion of the mesolimbic dopamine system. So normalized over the hypodopaminergic state, and we treated the psychological and physical withdrawal signs and improved the alcohol-seeking behavior. And next result is the medial prefrontal cortex and lateral habenula and lateral medial tegmental area pathway in the acupuncture in cocaine psychomotor response. This result were published in the addiction biology at 2021 and 23. This picture is the vibrating acupuncture instrument we developed to treatment of the acupuncture in the animal. So previously we researched that the acupuncture at the HD7 was significantly decreased the locomotor activity. The hyperlocomotor activity was decreased after the acupuncture stimulation. So when we treated the cocaine, significantly increased the locomotor activity, but when we treated the HD7 was significantly decreased of the locomotor activity after the cocaine injection. And this acupuncture signal was activated some specific neuronal sensory fiber, and this signal was converted from the spinal cord and dorsal column pathway. And actually, we detected about the mediation of the RMTG area on the acupuncture effect. So, and the first result showed that when we treated acupuncture at the HD7 was significantly decreased the locomotor activity induced by the cocaine, but gives some damage. Electrical lesion of the RMTG area will show that the acupuncture effect, the RMTG-X means the give some damage of the RMTG area. So when we treated acupuncture at the HD7, was showed the significantly decreased of the locomotor activity, but when we give some region of the RMTG area, it's not show the acupuncture effect. So this result mean that maybe the RMTG area was mediated the acupuncture effect in cocaine-induced psychomotor response. And next, we use the optogenetic technique. So inactivated the RMTG area. So when we treated the halorhodopsin and give some yellow light, so to make the inactivation of the RMTG area. So these results show that when we treated the HD7 was significantly decreased the locomotor activity, but a block of the RMTG area was disrupted this acupuncture effect. Yeah, this result show that. And next we check the specific neuronal type of which one is the mediated acupuncture effect. So GAD67 means the marker of the GABA neurons. So when we treated acupuncture, show that the GAD 67 and C4 submerged cell was significantly increased. And extracellular coding also showed that during the acupuncture stimulation was increased activity of the RMTG area. So it means that acupuncture effect is mediated, GABA neurons activity is mediated the acupuncture effect. And when we, without the acupuncture, just use in the chemically activated the RMTG area, it's also show that the effect of the decrease of the cocaine-induced locomotor activity is similarly in the acupuncture effect. So it means the mimic of the acupuncture effect when we chemically activated the RMTG area. And we detected the dopamine release, analyzed the dopamine release after the acupuncture stimulation by the in vivo portometry. So this research showed that the acupuncture stimulation significantly decreased the dopamine release in the nucleus accumbens, but when we inactivated the RMTH area, this acupuncture effect was abolished. And next, we checked the mediation of the prefrontal cortex. The experimental method was only in the before the experiment, and also showed that acupuncture effect was decreased locomotor activity, and region of the medial prefrontal cortex was abolished the acupuncture effect. And when we treated, when we detected the neuronal type of the from the medial prefrontal cortex to lateral habenula was a majority the glutaminergic neuron was mediated the acupuncture signal. And then we give the yellow light to block the medial prefrontal cortex and lateral habenula circuit by the optogenetic stimulation. This is to show that when we blocked the circuit, was the acupuncture effect was blocked it also. And next, we detected the dopamine release effect of the acupuncture. It also showed that when we treated the acupuncture was significantly decreased the dopamine release, but blocked of this circuit was abolished the acupuncture effect. So summarized, our results show that when we treated the acupuncture at the peripheral part of the acupoint, was activated some kind of sensory nerve. So, and this signal was conveyed from the peripheral acupoint to the brain. And this signal was acupuncture was activated the medial prefrontal cortex, and this signal was delivered to the lateral habenula by the glutaminergic neurons, and this signal also was conveyed to the RMTG area. And here, the acupuncture was activated the GABAergic signal, and it is mediated the dopamine release of the mesolimbic pathway. So, summarize that the acupuncture effect was in the drug addiction is mediated the various brain area, but is finally target of the mesolimbic dopamine system. Okay, thank you for your attention, and I finished my presentation.